This is a protein. Proteins are essential materials required for the cell to carry out activities necessary for its survival. In this presentation, we'll be covering the process as well as the different organelles that are involved in the creation of a protein.
In protein synthesis, there are two essential steps that are involved in the making. The first step we will be discussing is called transcription. The first step of transcription involves the DNA. Enzymes called RNA polymerase start the process of transcription by unzipping the DNA and inserting the RNA nucleotides into place. Cytosine and guanine nucleotides attract each other, and adenine and thymine nucleotides match up. But for the RNA, whenever a nucleotide is placed that is complementary to the adenine nucleotide on the DNA, instead of using thymine, the RNA uses a nucleotide called uracil. The messenger RNA or mRNA is then created in this process.
The copying of DNA starts and stops with the signal in the DNA. The RNA then exits the nucleus through a nucleopore. With this copy, the original DNA is still safe in the nucleus. This RNA searches for a ribosome. Ribosomes can be found on the endoplasmic reticulum or just floating around in the cytoplasm.
The RNA reads 3 at a time. Each section is called a codon. The ribosomes make the protein by reading the codon and assembles the correct amino acid using the transfer RNA. The transfer RNA transfers the right amino acid to build the protein. The transfer RNA has an anticodon which reads the codon on the messenger RNA and makes the specific amino acid to match that allows the translations to happen.
The second essential part is the translation. The mRNA now attaches to the ribosomes to start translating information to it. The tRNA anticodons match up to the codons of the mRNA. The next tRNA comes in and the amino acids form the peptide bond between the amino acids. The amino acids keep growing, forming into the protein. The protein keeps growing until the end of the codon.
When the protein is finished forming, it is now usable. The protein is now ready to be used. It is turned over to the endoplasmic reticulum. The vesicle transports the newly made protein to the Golgi body for its final packaging before being sent off in the vesicles to be sent outside of the cell membrane.